These are your notes on the structure of the cell membrane and the function of the different parts of the cell membrane. This is the overview of the cell membrane, so most of this information you should probably have to write down. A lot of this is new vocabulary. Anna, do you want to say hello to my kids? Say hi.
Hi.
Okay, so as far as the overview goes, the function of the cell membrane is that it separates the living cell from the non-living surroundings. It's very thin. It's 8 nanometers thick, so that's a very thin substance. Its function is that it controls traffic in and out of the cell, or it regulates the cell. It is said to be selectively permeable. That's a vocabulary word for you to write down. So what that means is that it allows some substances in more easily than others. And we've talked about that. We've talked about how if a substance is water soluble, it has to use a protein to get through the phospholipid bilayer because the tails are hydrophobic. But if it's lipid soluble, it's able to just diffuse directly through the membrane because it's made up of phospholipids. You should also know that it is made up of phospholipid proteins and other molecules such as carbohydrates and cholesterol.
Do the whole thing to make sure your speech is perfect.
We've talked a lot about phospholipids in class. If you don't have this information written down, you should write it down. And so this is what it looks like in terms of a chemical representation of a phospholipid. So it has the head is going to be hydrophilic, meaning water loving, and then the tails, so here are the tails on the chemical representation, are hydrophobic, meaning water fearing. And so the way that they orientate themselves is so that the heads, if this is inside of the cell and this is outside of the cell, then the heads are going to be exposed to the water that is inside and outside of the cell, which is fine because they are hydrophilic. And then the tails are hydrophobic, and so they are what actually regulate the cell and prevent water-soluble substances from diffusing directly through them according to their concentration gradients. So this is just a slide that shows you what the phospholipid bilayer actually looks like. So you can see the heads are outside and inside the cell, and then the tails are part of this bilayer section. And they will do this automatically. Sometimes part of the cell will pinch off or form around other molecules, and this phospholipid bilayer will automatically form like this. In 1992, Singer and Nicholson proposed that the membrane proteins are inserted into the phospholipid bilayer. This is when the whole idea of the fluid mosaic model came into play. We talked about that in class and how a mosaic is a piece of art that's made up of lots of different pieces that come together to form one art piece. That's the same thing for the cell membrane. It has this phospholipid bilayer. It's got all kinds of different types of proteins. And they all come together to form this one organelle called the cell membrane. So this is just showing you a bunch of different types of the cell, or the different parts of the cell membrane. We've talked about these in class. This is illustrated probably better than what I was able to draw for you. But here we have the extracellular fluid, so that's outside of the cell. Here we have the cytoplasm, and so that's inside of the cell. Whether it's outside or inside of the cell, it's water. And so we can see our peripheral proteins, which are proteins that are only located on one side of the cell. They don't go all the way through. And we have transmembrane proteins, which go across the membrane. So those are the ones that go all the way through. So you should write that down, and that's actually going to be coming up on the next slide, give you a better definition. But transmembrane proteins go all the way through. Peripheral proteins only go on one side. Cholesterol, we talked about how cholesterol is found within the tails of the cell membrane and how it helps to give it some structure. glyco, hopefully you'll remember that from our macromolecule unit when we were talking about glycogen. So this is the carbohydrate attached to a protein, and we talked about how, or I mentioned in class, how this is used for cell-to-cell signaling. So lots of different parts of this mosaic. So this is the slide that gives you the definitions of the protein, so you should write this information down. The proteins determine the membrane-specific functions, and so we've talked about how with all proteins it's their shape that determines their function, and that's the same thing here. The cell membrane and organelle membranes each have unique collection of proteins, so no two cells are going to be the same. If they're making up the same tissue, then they're going to have the same similar proteins, but their sequencing of proteins and stuff is going to be different. The different types of proteins, they can be categorized into two main groups. So you have your peripheral proteins, which again are going to be bound to one surface of the membrane, so they're going to only be on one side. They're not going to go all the way through. And they can have a couple of different functions, but one of them is as a surface identity marker, or that's referred to as an antigen. And so they help cells talk to other cells and signal the health of the cell that they are in. The second major category of proteins are called integral proteins, and these are transmembrane proteins, meaning they go all the way through the cell. So if you look at this picture right here, this would be a transmembrane protein, that would be a transmembrane or integral protein. They are generally going to be transport proteins, and so their function is going to be to get molecules from one side of the cell to the other side of the cell. And they can be channels. They could use, sometimes they use pumps. And so this would be an active transport, so it requires ATP. Carrier proteins are transmembrane proteins as well. So lots of different types of proteins, but two main categories: peripheral proteins, which are only on one side, integral proteins, or transmembrane proteins that go across the whole membrane. So you don't need to write these down, but this slide is just to give you a visual of the different types of proteins. And so these are going to be all transmembrane proteins, and so you can see that they're all going from one side of the membrane all the way down through the other side. And so we have a transporter protein. So it's just allowing for these red molecules to diffuse through it according to its concentration gradient. So here we have high concentration, here we have low concentration, and those molecules are able to diffuse from one side of the cell membrane to the other side of the cell membrane. This would be an enzyme type of protein. So we talked about enzymes when we were studying proteins. And so this substrate comes into the active site of the enzyme, it gets changed, and it gets released as a new molecule. This is a surface receptor, so it's able to receive molecules and then deposit them on the inside of the cell. This one down here is an identity marker, again, so it's got that glycoprotein attached to it, which helps in cell-to-cell identification and the cell being able to start an immune response if the cells need to. We know that when cells stack upon each other, they form tissues, and then tissues go on to form organs, and organs form our organ systems. And so the way that cells stick together to start to form these tissues is through cell adhesion. We talked about adhesion, and that's when things stick to other things. And so here we have one cell sticking to another cell. And then attachment to the cytoskeleton. So we haven't talked about cytoskeleton yet, but the cytoskeleton is kind of like the skeletal structure of the cell. And so it's what gives the cell a three-dimensional structure, and it allows so it prevents the cell from collapsing on itself. And so the proteins themselves will attach to the cytoskeleton to try to gain some support and structure for the phospholipid bilayer as well. This slide focuses on the membrane carbohydrates. So you need to know this, so you should write this down. The main role that they play, again, is, we've talked about this, is the cell-to-cell recognition. So allowing one cell to talk to the cell that's next to it, and say, okay, I'm a kidney cell, you're a kidney cell. We're going to adhere to each other with our adhesion proteins, and we're going to start to form some kidney tissue. The second thing, so that is important in organ and tissue development. And then another major thing that the that the carbohydrates do, or the glycoproteins do, is it sparks the immune system. And so if a cell comes in and its glycoprotein doesn't match the glycoprotein of a cell that's next to it, then that will start up your immune system. And so it's your body's way of identifying foreign cells, or cells that should not be in your body. And so if you have a tumor growing, for example, your body will start to identify that it's a tumor cell and not a kidney cell, because the glycoprotein that's attached to the tumor cell is not the same as the glycoprotein that's attached to your kidney cell. And so carbohydrates play a major role in cell-to-cell talking and just the overall health of the cell.
Bye, class. Oh, it's on.
Hold on, it's recording.
Welcome to science class. Now it is time for the head teacher to speak.
Today we're going to take notes on movement across the cell membrane. We've been talking about this a lot lately in class. Talking about diffusion, passive transport versus active transport. So we've defined diffusion many times before. If you are unfamiliar with that definition, you should write down what's in the yellow. But diffusion is the movement from areas high concentration, that should be an arrow, to areas of low concentration. It continues until equilibrium is reached. So this is equilibrium, when the molecules are spread out evenly, and there's no more concentration gradient. So this was actually an animation on the computer, but it doesn't work on the iPads. But it's just defining diffusion for you. It's a form of passive transport, which means that there's no energy needed. This is probably a redundant slide and nothing you need to write down. Water itself is able to diffuse through the membrane. So here we have high concentration, and here we have low concentration, and so water will flow in that direction. Remember that when we were talking specifically about the diffusion of water, we referred to it as osmosis. So osmosis is the diffusion of water across a selectively permeable membrane. So we know that the cell membrane is a boundary between the inside and the outside of the cell. It separates the cell from its environment, or the extracellular fluid. It cannot be impenetrable, meaning things have to be able to get in and out of it. Some of the materials that need to get in are food, carbohydrates, sugars, proteins, amino acids, lipids, salts, oxygen, water. No need for you to write those down, but you should be familiar with those. And other things that need to get out would be waste that's produced, ammonia, salts, carbon dioxide, water, and the products that the cell makes. And so the cell must be able to allow nutrients in and other products in, and allow for things to go out. So it cannot be impenetrable. We talked about this before, but molecules that can get directly through the membrane are fats and others lipids, because the membrane, again, is a phospholipid bilayer. And so molecules that are lipids can diffuse directly through there. Other molecules that are large or charged or polar cannot. And so what they're going to have to do is use a protein to get across the phospholipid bilayer. And so again, this was an animation on the computer, but not working on the iPads. So here's our bilayer with our heads that are hydrophilic facing outward, and our tails that are hydrophobic facing inward. So the membrane is said to be semi-permeable. We've also referred to it as selectively permeable. And so what that does is it means that it allows some things to get through it and others not. If you don't know what that definition is, you should write that down, but otherwise we could probably just move on. And so if materials cannot diffuse directly through the phospholipid bilayer because they are ions or salts or large molecules, then they need to use protein channels or protein carriers. And so they need to use these transport proteins. These are referred to as transmembrane or integral proteins, and they go all the way through the membrane. So you can see how it goes all the way through the membrane. It does not require energy because it's going with the concentration gradient. As long as the molecules are moving with the concentration gradient, it does not require energy. So going from high to low. Passive transport, no energy, but you can still use these proteins. This one's showing you a channel protein, which I've said is just kind of like a tunnel for the molecules to bypass that phospholipid bilayer. Active transport, the cells need to move, when cells need to move molecules against their concentration gradient, they can do that. It's going to require the use of a carrier protein. So here's our carrier protein. You can see that it's moving it from high concentration to low concentration here. This is actually not a very good picture, so I apologize for that. So ignore this and ignore this. But it's moving it from low concentration to high concentration. So the molecule would go in, it would find itself compatible with a protein, and that protein would release it on the other side. These are sometimes referred to as pumps, protein pumps, and again, it costs energy. The energies that cell use is ATP. There's different types of active transport. We don't really need to concern ourselves with those, but there's symport, in which it happens simultaneously. So molecules would go on this side, and molecules would go on this side, and they would flip-flop, and so they'd be exchanged on either sides. Sometimes they work on, or that's antiport, and then symport is when they work together, and they're going in the same direction, so they're both released on the same side, so that's the sym, and then versus the antiport. This is the information you should write down. So, if you're talking about passive transport, not going to use any energy. There's simple diffusion, which is the diffusion of molecules directly through the phospholipid bilayer, going with the concentration gradient, so from high to low. There's facilitated diffusion, which is the use of a protein channel or a carrier protein, again, going with that concentration gradient. This is how the majority of molecules get in and out of the cell. Active transport is the use of energy. It's diffusion against the concentration gradient, so it's going from low to high. It's going to require the use of a protein carrier and energy, and the energy that's used is ATP. So this is the information you should write down. This is just giving you a visual of the different types of transport. So we have simple diffusion straight through, facilitated diffusion using a protein, and active transport going against that concentration gradient. So here you can see it's low concentration going to high concentration and requiring that use of of ATP, and that's what makes it active. We talked about endocytosis and exocytosis. So endocytosis is just the taking in of large molecules. And so this picture kind of works in this is exocytosis. So here we have a vesicle that's already been formed. It comes up, the membrane of the vesicle reconnects with the phospholipids and releases its substances into the extracellular fluid. This would work in the reverse of endocytosis, so just put the arrows going in the other direction really, and it becomes endocytosis. So molecules come in, this pinches off, vesicle forms, and is moved into the cell. So endocytosis is the taking in of large molecules, and exocytosis is the expelling or letting go of large molecules. You need to know those two vocabulary words. Nothing to write down on this slide, just giving you a visual of the different types of endocytosis, but we're not going to be required to know those. So we've talked about how water is different and that it is referred to as osmosis. And this is going to start talking about those different types of solution types. Again, osmosis is the diffusion of water. Water is very important to life, so we talk about it separately. It's going to go from high to low across the semi-permeable membrane. This is just giving you a example. And I like this slide because it shows you hypotonic versus hypertonic side by side. So hypo, we said, makes the cell blow up, and there's other slides for you to write down this information, but this is just a good visual. And hyper, that E, means exit. And so when we're talking about what those mean, it means that we have more water than solute, and then here we have more solute than water. And so we'll continue on with those until it reaches equilibrium. So this is going to be your dynamic equilibrium, which means that there's no more concentration gradient. So again, just giving you this visual hypertonic, hypotonic, and isotonic. I find it easier to remember it in terms of the water movement, and then just knowing that the solute is the opposite of that, right? So if you have less water, you're going to have more solute. If you have more water, you're going to have less solute. And then isotonic is equal. So I talk primarily in terms of just the water because I think it's easier to just remember it in one way and then just apply it to the solute. Again, just giving you some visuals of what cells do when they're placed in these different solutions. So hypotonic, we said, makes the cell blow up. So here's our cell blowing up. So water is continuing to flow in. The cell membrane can't withstand the pressure and it can burst. This is a plant cell, and so plant cells are happy. Okay, you can see that it says the normal condition for a plant. You can see the cell membrane pushing outward against the cell wall, but the cell wall is preventing it from bursting. Isotonic solution, this is normal for an animal cell, and a little bit flaccid for a plant cell. Plants like to have hypotonic solutions. And then hypertonic solutions are not good for either one. You can see how the animal cell is all shriveled up. And this is called plasmolysis or plasmolysis. And so what happens is the cell membrane actually pulls inward away from the cell wall, and that cell would not function properly. So different things happen for animal cells compared to plant cells when they're placed in the different solutions. So for isotonic, you need to know that there's no net movement of water. Now that doesn't mean that water is not moving in and out. It just means that it's doing so evenly. So you can see there's water coming in and there's water going out. So there's no concentration gradient. And so there's no movement of water in one particular direction. So animal cells like this, plant cells not necessarily. But just you need to, the important thing, like I said, is to know that in an isotonic solution, there's no net movement of water. It moves in equally in both directions. Hypotonic cell, animal cells don't do well with hypotonic solutions. We've said that the water will flow in until it can burst. This was that question on our homework a couple nights ago with the paramecium. And so we said that the paramecium is placed in fresh water. What's going to happen to it? And so the water is going to continue to flow into the paramecium until it bursts, or it has this contractile vacuole, which is inside of it, and it's actually a picture of it on the next slide. And so what it does is it pumps water out of the cell, but that requires energy because it's going against that concentration gradient. And so that's what that question was referring to on your homework. Plant cells love this. This is when they're doing their best. Just a reminder that hypo makes the cell blow up. I think it's an easy way to remember that in terms of the water movement. This is that picture of the paramecium. So here's our paramecium. Hopefully it looks familiar to what you found under the microscope. So here's our contractile vacuole. So you can see how in this paramecium, it's being submerged in fresh water. So water is flowing in through the membrane of this unicellular organism, or protist. And so what's happening is that this contractile vacuole is starting to swell. And then once it gets to the point where it's reached its maximum size, it will literally contract and force the water to leave the paramecium so the paramecium doesn't explode, essentially. So that's what happens when you put a paramecium in fresh water. This, though, requires ATP because it's active transport going against the concentration gradient. Last one is hypertonic solutions. Neither plant nor animal cells are happy with this. So it's a cell in salt water. Okay, you can't take in salt water. You can't live off of salt water because it's going to force all of the water to flow out of your cells, and your cells will shrivel up and die. So this is movement. Water exits, E equals exit.